Alright guys, welcome back to Underground Science, and in this video we'll pretty much be picking up from the last video for the interaction between a macrophage B cell and a CD4 positive cell, or also called a helper T cell. So I got a quick screenshot of our last video, so we can go ahead and do a quick review and then continue and finish the basic interaction off, alright? So let's go and get started. So we have our major histocompatibility complex 2 right here of our macrophage. So let's go and label that. So this is our macrophage. All right, so that's our macrophage. And we initially, what we did was we just put the antigen on and assumed that we had broken the pathogen down into smaller pieces, right? So just remember, like the macrophage phagocytoses the pathogen, right, as a phagosome. And what it does then is that it actually breaks it up via lysosomes, or lysozymes, I should say, and into smaller antigens, right? So in this case, we should draw orange. We should draw it as orange, right? And so right here as a whole, right, all of these smaller antigens inside this little vesicle, right, is called a phagosome. So this whole vesicle is called a phagosome. All right, so let's just go and write this quick, because we already know the process of phagocytosis and the process of breaking it down via lysozymes, and then these antigens get presented, and then we talked about how this helper T cell here, so it kind of got cut off, but this is a helper T cell. All right, and this helper T cell was inactive previously, but then we ended off saying that once this helper T cell actually comes along here, and if the T cell receptor, so this right here is the T cell receptor. All right, so we have to keep on relabeling things so we stay on the same page, right? So this T cell receptor, if it matches this particular complementary antigen, then it'll bind there, successfully bind. Then we say we need a CD4 glycoprotein that's present on helper T cell to also bind to the MHC2 by itself, right? Then this process results in the release of interleukin-1, we said. All right, so after this binding process, right, we see interleukin-1 release from the macrophage. This interleukin-1 can then activate this helper T cell. Previously it was inactive. It can activate this helper T cell, and then this helper T cell is now activated with its T cell receptor, and then we said we'll pick up in this video. So next, what it can go do is pretty much it can go find a B lymphocyte that also has this same exact antigen presented on its surface of its membrane via a MHC2 complex. So let's see what I mean by that. So let's say our helper T cell right here is active as crap, right? And it's going to go find a B lymphocyte. Let's say we have a B lymphocyte chilling over here, right? It's already got, or it's ahead of the game. It has the same exact antigen, because remember, this helper T cell has a T cell receptor only for this specific complementary antigen. So only this specific antigen will fit the T cell receptor once it binds. So this T cell, this activated helper T cell, now recognizes that specific antigen because it got active. It was the helper T cell became active when our macrophage released interleukin-1, right? So now it's going to go find a B lymphocyte that has the same exact antigen as our antigen over here, right? So let's say it finds a B lymphocyte that also has the same exact antigen that represented by orange, presented on its MHC2 complex. So let's say this is our MHC2 complex, and we said that this is a B lymphocyte, right? So our B lymphocyte is waiting, because remember, our B lymphocyte needs helper T cells in order to proliferate. So our helper T cell will come here and be like, oh, sweet, right? I saw you. I got that's the same antigen. I'm activated for that specific antigen. So the helper T cell will actually bind here. So what we'll see, right? So let's go and draw this as a sequence of events. So then what we'll see is that this B lymphocyte will then have so let's go and draw the MHC2 again. Let's go draw its antigen, and then we'll see the T lymphocyte, the helper T cell, has now successfully attached itself, right, via its T cell receptor, its T cell receptor, right, onto what? The MHC2, right, or not the MHC2, specifically the antigen on the MHC2 complex, all right? However, if we label this part, this is going to be the MHC2 complex. And then we have our CD4 glycoprotein that's here, right, that attaches itself on the MHC2 complex. And so let's go and label that on our T cell, our help on, I can't speak, sorry about that, on our helper T cell. This is our CD4 glycoprotein, and this is our helper T cell. All right, so I'm going to write that a little neater, our helper T cell. All right, so now that it does this, what's going to happen is that our helper T cell, once it binds to it, so remember this is also our B lymphocyte, so it's also going to have membrane-bound antibodies, right, on its surface, on its cell membrane. And these membrane-bound antibodies also can have bigger versions of this antigen, right, as a whole even, of the pathogen bound on itself to, it can tag it, right, and attract other white blood cells to this area of where the B lymphocytes are. So, but anyways, right, if we look at this part, once our helper T cell has now successfully attached itself on this antigen presenting part via our CD4 glycoprotein on the helper T cell and via our T cell receptor on the helper T cell, then what's going to happen is that then our helper T cell, right, can then go even more beast mode and start releasing some lymphokines and cytokines, so some inflammatory markers. So it can then start releasing some lymphokines and cytokines, right? So inflammatory markers that can then travel over here and attach onto the B lymphocyte, and it can trigger then the B lymphocyte to start proliferating into the two main types of really important B lymphocytes that are going to be our effector B lymphocytes, so our effector B cells, and our memory B cells. All right, and if you guys remember, another name for the effector B cells are also plasma B cells. All right, these plasma B cells are pretty much just going to start producing a bunch of antibodies. All right, those antibodies that are the same exact antibodies as the one that was on the surface of the B cell's membrane, so that it can go and find other same exact of these pathogens and go and opsonize, right, perform opsonization and attract more white blood cells to destroy these pathogens. That's somewhere else in the body, right? All right, so that's going to happen because of lymphokines and cytokines released by the helper T cell once we have that binding process that takes place, all right? But also what can happen is that our B lymphocyte can also in turn, right, release its own cytokines that will actually help the helper T cell proliferate. So we also see, so let's go and draw this in, let's see, I guess green, right? We also see that once our B lymphocytes start getting activated and start proliferating, these B lymphocytes can also release some cytokines, right? Can also release, so these are some cytokines released by the B lymphocytes, can also release specific types of cytokines that can then travel over, it's going to be kind of weird, sorry about that, can then travel over to the helper T cell. And then once these cytokines bind to the helper T cell, these cytokines can cause the helper T cell to start proliferating into two main types of helper T cells. So one main type that the helper T cells will start proliferating into are memory helper T cells, and our effector helper T cells, right? And our effector helper T cells. And so these two, right, so our memory helper T cells and our memory B cells will sit out for another time when the same pathogen comes back again. Then we'll actually get rid of this pathogen in a future exposure much faster because of our already made memory helper T cells and our memory B cells. We can activate this process much faster, all right? And so what the effector helper T cells are going to do is that it's going to go find more B lymphocytes that have this specific antigen, right? That have the specific antigen presented on its MHC2. And it'll go find more B lymphocytes and do the same process and keep on triggering or inducing the proliferation of B cells into memory B cells and effector B cells. And we're going to have more proliferation of memory helper T cells and effector helper T cells, right? And another thing that I want to mention, guys, is that these effector helper T cells, so pretty much extra helper T cells, as a whole, release a really important cytokine called interleukin-2. Sorry about that, interleukin-2, right? So as a group, they start releasing a cytokine called interleukin-2, which is a really important cytokine, right? They also release other cytokines, but interleukin-2 is a really important one because interleukin-2 can actually go to nearby T lymphocytes, and what it can do is that it can induce the proliferation of nearby T lymphocytes into T cells like cytotoxic T cells, which are important for taking care of cells that are infected or cells that are cancerous. And it can also, interleukin-2 can also stimulate or induce the proliferation of T lymphocytes into another type of T cells called memory T cells, or memory helper T cells, I should say. All right. So as we can see, interleukin-2 is really important. Okay. And so again, this will help all together, and also this one will help all together to improve our immune response and take care of whatever pathogen that's trying to destroy us. All right. So that's pretty much what I wanted to get through in this video. Sorry about that random mark. I just want to talk about the basic interactions between macrophages, B cells, and CD4 positive cells, or also known as helper T cells. Just remember that we have interleukin-1 released by our macrophage that's mostly responsible for activating our helper T cell. That can then go find the same exact antigen on a B lymphocyte. And after our helper T cell finds that same exact antigen, it can bind there with the help of the CD4 protein. Then we know that the B lymphocyte and the helper T cell can together release cytokines on each other and lymphokines, right? Especially the T helper cell can release lymphokines and cytokines. The B lymphocyte can release cytokines. And all these inflammatory markers can help the helper T cell proliferate into memory helper T cells and effector helper T cells. And all these cytokines and the lymphokines also released by the helper T cell can help the B lymphocytes, right? So the lymphokines released by the helper T cell can induce the proliferation of this B lymphocyte into effector B cells, or also known as plasma B cells, and memory B cells. And then we said all these, the groups that are the cloned helper T cells as a group can then release interleukin-2 and other cytokines. However, interleukin-2 is really important because we said that it can actually activate nearby T lymphocytes to proliferate into cytotoxic T cells and memory T cells. All right, and we'll talk about cytotoxic T cells more in a future video. All right, but it's mostly important for cells that are infected, right? Cells that are infected and cells that are cancerous, to take care of those cells, all right? Cells that are cancerous. And so, yeah, I hope this video made sense to you guys. I hope to see you guys in my future videos where I'll talk about more cool topics about biochem, physics, and chem. Don't forget to hit that notification bell, like, and subscribe, and see you guys later, and stay safe.